Okay, so welcome to this next video in which we're discussing nitric oxide as the endothelium-dependent hyperpolarization factor, the EDHF. Okay, so we are in the process of discussing the experiments that Chen et al. Did in 1988 to show that nitric oxide was not mediating this hyperpolarization that you see of the electrical potential difference across smooth muscle cell membranes in response to stimulating the endothelial cells with acetylcholine. Okay, so what he did was he used hemoglobin, because hemoglobin has these four phosphatic heme groups which have this free sixth coordinate bond, and nitric oxide can bind to this free sixth coordinate bond on these phosphatic heme groups. So effectively hemoglobin can mop up the nitric oxide. Okay, so what you do is you expose these endothelial cells to lots of hemoglobin to mop up the nitric oxide and prevent the nitric oxide going to the smooth muscle cells. And basically what you see is that if you then stimulate these endothelial cells with acetylcholine or carbachol in the presence of the hemoglobin, which will stop any nitric oxide getting between the endothelial cells and their smooth muscle cell, you still observe the hyperpolarization of the electrical potential difference across the smooth muscle cell membrane. So you still you can still see endothelium-dependent hyperpolarization even when nitric oxide cannot possibly be moving between the endothelial cells and the smooth muscle cells. In addition, in 1988 as well, Taylor et al. Taylor et al. Showed that if you use the nitrovasodilator drug sodium nitroprusside, often abbreviated to SMP for short, so I'll write out its full name at least once, sodium nitroprusside, then basically what this drug does is that it undergoes a chemical reaction and releases nitric oxide as a product of this chemical reaction. So when you put it into the blood vessel, what it's going to do is it's going to undergo this chemical reaction, release nitric oxide without stimulating the endothelial cells. This nitric oxide will then go to the smooth muscle cells in the wall of the blood vessel. And if nitric oxide was responsible for causing this hyperpolarization, you should measure a hyperpolarization of the smooth muscle cell membrane, electrical potential difference, okay? Only you don't. So those were two quite convincing experiments that showed that nitric oxide didn't seem to have anything to do with endothelium-dependent hyperpolarization, which Bolton had observed in 1984. Okay, right. So now let's discuss the experiment that seems to show otherwise. Okay, so we'll start then with Tarr et al. In 1990. Okay, so Tarr et al. Okay, so Tarr et al. In 1990 performed some experiments which appear to contradict this and show how nitric oxide, after all, does seem to be quite involved in the endothelium-dependent hyperpolarization. Okay, right. So let's describe the experiments that Tarr et al. Did. Well, basically, what they did is they took uterine arteries, okay, from guinea pigs. So they used the uterine artery from guinea pigs. Okay. Uterine artery from guinea pigs. And what they had was uterine arteries from guinea pigs which were pre-constricted. So I need to explain this because this may well be the reason that these two experiments give such different results, because Tarr et al. Is basically going to show that nitric oxide is responsible for this depolarization. Okay, right. So, this hyperpolarization, at least in part. Okay, so what Tarr et al. Did was they took uterine arteries from guinea pigs and they exposed them to noradrenaline. Okay, so they exposed them to noradrenaline. Now, what will noradrenaline do to the smooth muscle cells of the blood vessel? Okay, so they're going to bathe the smooth muscle cells of this uterine artery in noradrenaline. Now, what noradrenaline does is it acts on the alpha-1 receptors which are in the smooth muscle cells. So let's draw this out here. Okay, so it's going to, let me draw a smooth muscle cell here. It's going to act on the alpha-1 receptors on this smooth muscle cell. So here's our alpha-1 adrenergic receptor on our smooth muscle cell. So this is the alpha-1 receptor. Okay, so noradrenaline, or norepinephrine, as its new name is, so I'll write it out. Is it going to fit in? Nephrin? Right, okay, so norepinephrine basically is going to cause contraction of this smooth muscle cell, and it's going to act through the alpha-1 receptor, so I'll colour this in. So here's the alpha-1 receptor in pink. Now the alpha-1 receptor is coupled to a GQ heterotrimeric G protein. Okay, so let's draw that here. So heterotrimeric G proteins consist of an alpha subunit, a beta subunit, and also a gamma subunit here. Okay, and if this G protein, this heterotrimeric G protein, is a GQ heterotrimeric G protein, it means that the alpha subunit has to be alpha Q. Okay, so there are 16 different alpha subunits you can use in a heterotrimeric G protein. In addition, there are 5 beta subunits you can use, and there are 12 gamma subunits. Now, the name of the G protein does not tell us what the beta and the gamma subunits are, so all that's important for the naming of this G protein is the alpha Q subunit. Now, alpha Q, in its off state, is bound to guanosine diphosphate. Okay, so GDP here. Now, basically, when noradrenaline binds to this alpha-1 receptor, what's going to happen is the alpha-1 receptor is going to become catalytically active, and it's going to chop the GDP, the guanosine diphosphate, off of the alpha-Q subunit, and instead it's going to find a GTP, a guanosine triphosphate molecule in the cytoplasm of the cell, and it's going to bind that onto the alpha-Q subunit instead. So, what you're going to get is alpha-Q subunits which have GTP bound to them, i.e. alpha-Q subunits which are in the on state. Okay, and this complex here, this protein, is often known as the alpha-Q GTP subunit. Okay, and once the alpha-Q subunit has GTP bound to it, it no longer wants to associate with the beta and the gamma subunit, so they go off. They remain bound with each other, so I'll show this happening. So they remain bound with each other here, so here's the beta subunit and here's the gamma subunit, but they no longer combined with the alpha-Q subunit. Okay, so let's go over the page and continue this pathway. Okay, right. So, what does this alpha-Q GTP subunit do? So if this is our alpha-Q subunit, and it's bound to guanosine triphosphate, GTP here, I'll colour it in a nice orange colour. Well, it basically is going to activate an enzyme in the phospholipid bilayer of these smooth muscle cells. So it's going to activate an enzyme known as phospholipase C-beta. So if this is the phospholipid bilayer of our cells, then within the phospholipid bilayer there is an enzyme known as phospholipase PLC-beta. Okay? And this is sitting in the phospholipid bilayer. So this PLC-beta is short for phospho, that's the P, lipase, that's the L, and then C, that's the C, and then beta. So let's have phospholipase C-beta in a colour. So what's going to happen is the alpha-Q GTP subunit is going to come and bind to the phospholipase C-beta enzyme, and it's going to activate it. And now, once the phospholipase C-beta enzyme is activated, it's going to act on a lipid molecule that is within phospholipid bilayers. Okay, so there is a lipid molecule within phospholipid bilayers that is called PIP2. So, within the plasma membrane, you have this molecule, and basically it's just a modified phospholipid. So PIP2 is within the phospholipid bilayer, and it's just a modified phospholipid. So it takes the place of a normal phospholipid. And what phospholipase C beta is going to now do, now that the alpha-Q-GTP subunit has activated it, is it's going to take this molecule and it's going to break it down basically into two new molecules. So it's going to break it down into diacylglyceride, abbreviated to DAG, over here, which will remain in the phospholipid bilayer, and then it's going to break it down into another portion. It's basically going to chop off the head of the PIP2. It's going to chop it into another portion, which is inositol 1,4,5-trisphosphate, IP3 over here. Okay, right. So, basically what happens is when you activate the phospholipase C beta, it takes PIP2, which is a component of the phospholipid bilayer, and it breaks it down into diacylglyceride and IP3. The IP3 then acts on IP3 receptors in the membrane of the sarcoplasmic reticulum. Okay, so this is going to go off and activate receptors for IP3, which are in the membrane of the sarcoplasmic reticulum. So here is our intracellular organelle, which is the sarcoplasmic reticulum, because we are talking about smooth muscle cells. So in any sort of muscle cell, the endoplasmic reticulum is renamed the sarcoplasmic reticulum. So this is the sarcoplasmic reticulum. And basically, IP3 is going to prime these IP3 receptors for opening. So it doesn't actually open the IP3 receptors itself, but it binds to them and then exposes binding sites for calcium. And if calcium binds to those binding sites, the IP3 receptor will then open. So it takes the IP3 receptor and gives it the ability to open, basically. It doesn't open it itself. Instead, it means that the IP3 receptor can open, whereas without IP3 bound, it can't open. Okay, so what will happen is some calcium will bind here, because there is some calcium in the cytoplasm of the cell, and then what will happen is you'll get the release of calcium here from the lumen of the sarcoplasmic reticulum. And what this will cause is it will cause two things. Okay, one, it will cause contraction. Okay, and I don't know why I put this below, but it will cause contraction of the smooth muscle cell. Okay, two, it will cause depolarization, and I want to explain to you why it will cause depolarization. Basically, what are you doing? You are moving calcium ions out of the SR into the cytoplasm of the cell. Okay? Calcium is a positively charged ion. Okay? So when you do this, what are you going to do? You're going to raise the electrical potential of the intracellular compartment. So this is going to go up because you're bringing calcium ions in. Now, you're not going to do anything to the electrical potential of the extracellular compartment, because that's not being affected at all. However, when we think about the electrical potential difference from extracellular to intracellular, okay, this is the electrical potential difference intracellularly minus the electrical potential extracellularly. It's how much lower the intracellular electrical potential is compared to the extracellular electrical potential, or how much different it is from the extracellular electrical potential. So usually this is negative 60 because the intracellular electrical potential is 60 millivolts lower than the extracellular electrical potential. However, if we raise the electrical potential intracellularly, then the amount by which the intracellular electrical potential is lower than the extracellular electrical potential will decrease, i.e. this number will go from being something like negative 60 millivolts to being a less negative number, maybe negative 50 millivolts. Okay, and that phenomenon of the difference between them becoming less is known as depolarization. So you're losing the polarity in electrical potential across the membrane. Okay, now the astute ones amongst you will notice that there's a problem with this. because you're moving the calcium out of the SR. So, if we look at the electrical potential of the SR, this is going to plummet, basically, because the calcium is being moved out. So you're moving positive charge out. So the electrical potential of the SR is going down, and the electrical potential of the intracellular compartment is going up. Okay, so what's going to happen? You're going to create an electrical potential difference between the intracellular compartment and the sarcoplasmic reticulum. This intracellular electrical potential is going to now be greater than the electrical potential of the sarcoplasmic reticulum. Now let me stress, usually within a cell, so let me draw a cell here, usually the electrical potential of the cytoplasm here, let's say EI here, and then the electrical potential within the sarcoplasmic reticulum, which is this blob inside, these are usually the same, i.e. they are electrically neutral. There is no electrical gradient across the membrane. However, when you move this calcium, what you're going to start building is an electrical potential difference. Now, what would it happen if we just kept letting this happen? Well, what would eventually happen is you'd stop moving calcium, because the electrical potential difference would become so large that the calcium just cannot move out of the ESR. Even though the concentration gradient is pushing calcium out, you just simply can't move it, because the electrical potential gradient is opposing the concentration gradient. So the point at which the concentration gradient favouring movement of calcium out is balanced by the electrical gradient favouring calcium movement in, that would be your point where you've got no net movement of calcium across the membrane anymore. However, something comes to the rescue of this and stops you ever getting to an electrical potential difference that would cause that. Basically, these IP3 receptors are also permeable to potassium ions. So potassium can basically move in the opposite direction. Okay, so when you start to build up an electrical potential difference between these two, what will happen is the potassium will start to move into the SR. And I want to stress the potassium concentration in the cytoplasm is the same, roughly, as the potassium concentration in the SR lumen. So there is no concentration driving force driving potassium in. Instead, potassium will only start to move in once there is an electrical potential difference between the two compartments. Once the SR lumen is at a lower electrical potential than the intracellular compartment, potassium will start to move in, and that will start to act as the counterion for calcium and potassium. Now you might say, well, surely this is perfect. This will stop you ever building up an electrical potential difference. No, it won't. Okay, because the potassium is only moving in because of the existence of the electrical potential difference. So you will establish an electrical potential difference first, and then you'll get the potassium current coming in. Yes, if you left it for infinity amount of time, then eventually the potassium would neutralize this gradient. Of course it would. But the reality is that the potassium movement is slow through this channel, so what will happen first is you'll build the electrical potential difference, and the potassium will just be chasing, trying to neutralize this, and at the same time calcium will still be coming out, so it won't remove all of the electrical potential difference. So the point is that you do build up an electrical potential difference across this membrane, but it never gets to the point where it would actually stop the movement of calcium, because before that potassium starts moving in and acting basically as the counterion for the movement of calcium and stopping the building up of the electrical potential any higher, basically. This means that you do raise the electrical potential of the intracellular compartment, and that's why you get a depolarization of the membrane, of the electrical potential difference across the plasma membrane in response to noradrenaline. Okay, and we'll continue this discussion of what Ter et al. Did in the next video.